The aim of this video article is to demonstrate how to quantify the frequency of bona fide neural stem cells in a mixed population of neural precursor cells using the Neural-Colony Forming Cell Assay. This assay can be used for primary or cultured neural precursor cells from adult or embryonic CNS. Harvested single cells are plated at an appropriate density and then incubated for 3 weeks in a 37-degree incubator. The frequency of neural progenitor and bona fide neural stem cells is calculated respectively by counting the number of colonies that are less than 2 millimeters and greater than or equal to 2 millimeters.
Using the Neurosphere assay to determine the frequency of bona fide neural stem cells in a mixed neural cell population overestimates the number of neural stem cells. Because there is not a one-to-one relationship between neurosphere formation and neural stem cells. We developed the Neural-Colony Forming Cell Assay in our lab to overcome this limitation. This assay can easily discriminate bona fide neural stem cells from progenitor cells based on their long-term proliferation potential. Today we show you how to use this semi-solid collagen-based assay to enumerate the frequency of neural stem cells in cells prepared from dissociated neurospheres from E14 mouse brain. Okay, let's get started.
For this assay we need NeuroCult neural stem cell proliferation supplement, basal medium, Neural-Colony Forming Cell Assay serum-free medium, and collagen. Pipette 1 ml of complete neural stem cell medium into a 15 ml falcon tube. Take as much volume from the cell suspension you have already prepared and pass through a 40 micron mesh filter in order to reach a concentration of 2.2 times 10 to the 5 cells per ml. To plate cells in duplicate in two 35 mm culture dishes, mix 1.7 ml of NeuroCult Neural-Colony Forming Cell Assay serum-free medium without cytokines with 330 microliter of NeuroCult Neural Stem Cell Proliferation Supplement, then add 6.6 microliter of EGF, and 32 microliters of penicillin streptomycin. Add 25 microliter of cell suspension to the prepared medium. Add 1.3 ml of cold collagen solution to the cell suspension and mix well, but gently to avoid introducing air bubbles. Then dispense the mixed solution to the center of each culture dish, approximately 1.5 ml per dish, and let the mixture spread evenly over the surface of the dish. 3 ml of sterile water is added to an open 35 mm dish placed alongside the culture dishes in the large Petri plate. This helps maintain optimal humidity during the incubation period. Label the plate and transfer to an incubator set at 37 degrees Celsius, 5% carbon dioxide and 95% humidity. It is important not to disturb the culture during the first hours so that the collagen can congeal. To feed the Neural-Colony Forming Cell Assay culture, take 200 microliter of complete neural stem cell medium and add 10 microliters of EGF at a concentration of 10 micrograms per ml. Add 60 microliter of the complete NeuroCult replenishment medium to the center of each dish once every 7 days during the entire incubation period. Each culture dish is placed on a gridded scoring dish of grid size 2 mm by 2 mm and then placed on the microscope stage. Using low-power objective lens, each dish is scanned and colonies are scored based on their size. Here are representative results of the assay at the end of the first week. At this time point, neural stem and progenitor cell-derived colonies are indistinguishable. After 3 weeks, colonies can be classified into two major categories: those less than 2 mm in diameter, which are progenitor cell-derived, and those greater than or equal to 2 mm in diameter, which are stem cell-derived. The colonies that are greater than or equal to 2 mm in diameter fulfill all the functional criteria of a bona fide neural stem cell.
In the colony forming cell assay, plating cells with an appropriate density is very important. Having too few colonies keeps the rare neural stem cell colonies under detection levels. On the other hand, too many colonies will result in overcrowding and depletion of gross medium components. Thank you for watching and good luck with your experiment.